Hello and welcome back to the last part of the meiosis, and after this we are going to the part of the discussion of the questions. Now in the previous session we have already discussed regarding the prophase 1 of meiosis 1, and with an elaborate part we have discussed regarding all the 5 substages of prophase 1, where we have studied regarding leptotin, zygotin, pactin, diplotin as well as the dikinesis part. Now let us continue the part of the meiosis. So we are at the part of the meiosis 1 where we have completed only prophase 1 of meiosis 1. The second part is the metaphase 1 of meiosis 1 in the chapter cell cycle as well as cell division. So here we have the meiosis 1. The rest part of the meiosis, the second stage is regarding metaphase 1, followed by the Anaphase 1 and Telophase 1. Okay. Now in the prophase, the chromatin thread gets condensed to form into chromosomes, and now these chromosomes we call them as bivalent chromosomes. They get aligned at the equatorial plane, right? Then the centrioles will move towards the opposite poles. They produce the spindle fibers. These spindle fibers will attach to the kinetochores of what? Kinetochores of the chromosomes, and 2 metaphase plates are formed. So how the metaphase plate is formed? So let me explain you. So in the metaphase 1, if we look at the structure, now here chromosomes are arranged at the equatorial plane. Now all the chromosomes are arranged at the equatorial plane, forming 2 metaphase plate, because the homologous chromosomes are aligned at the center. They are present at the center. We call them as homologous chromosomes. Iverdu jodi jodi. Hinge ಎಷ್ಟು chromosomes irtavu ಅಷ್ಟೇ pair ಆಗಿ ಇರ್ತಾವು. Okay, they are arranged at the equatorial plane in this way, right? Now here we have the centrioles where it produces the spindle fibers. These spindle fibers will attach to the kinetochores of each of these chromosomes. Okay, so we call them as bivalent. Bivalent andre ಎರಡು ಇರ್ತಾವು, and they are present in tetrad form. Tetrad form ಒಳಗಡೆ ಇರುವಂತವು. Okay, now what happens is in the metaphase 1, so explanation is very simple as already we explained in the part of the meiosis. Exactly same, but little change is there. That change is metaphase is a condition where all the homologous chromosomes are arranged at the equatorial plane. The centrioles will move towards the opposite poles, producing the spindle fibers. These spindle fibers will attach to the kinetochores of the chromosomes. Okay, then after that, this is the characteristic feature of the metaphase 1. I am explaining you along with the diagram, so you have to note down the characters, right? Then after the metaphase 1, it is followed by anaphase 1. So after the metaphase we have anaphase. So what happens in the anaphase? Anaphase is a condition where the spindle fibers will contract, they will pull the chromosomes, and the chromosomes start moving towards opposite poles. But the thing here is the total number of chromosomes which are arranged here is 23 pairs, 23 ಜೋಡಿಗಳು ಇರ್ತಾವೆ, where we call them as 46, 46 ಇರ್ತಾವೆ. Out of these, half of the chromosome will move towards one pole, half of the chromosome will move towards the other pole. ಅಂದರೆ ಅರ್ಧ ಅರ್ಧ 46 ಒಳಗಡೆ ಏನು ಕ್ರೋಮೋಸೋಮ್ಸ್ ಇರ್ತಾವ ಇವು ಅರ್ಧ ಅರ್ಧ ಮೂಮೆಂಟ್ ಆಗ್ತಾವೆ. then we call that as a what? Dyad. The dyad chromosomes will move. ಇಲ್ಲಿ ಟೆಟ್ರಾಡ್ ಯಾಕಂದ್ರೆ 4 ಆರ್ಮ್ಸ್ ಅದಾವೆ, 1 2 3 4 chromatid arms ಇರುವಂಥವು. This is a tetrad condition. Now in the anaphase they become dyad. That means the chromosomes starts moving towards the opposite poles. Look at this. This is called as a dyad. The centromere does not split. Remember, the centromere does not split. The chromosomes start moving towards opposite poles. Half of the chromosomes will move towards one pole and other half to the other pole. Right? We can call it as north pole and south pole. ಎರಡು polar ends ಕಡೆ movement ಆಗ್ಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗ್ತಾವೆ. So this is how the movement of chromosomes occurs at the anaphase. That's why this is called as what? Reductional division. Right? Reductional, because half of the chromosomes are found in each daughter cells. 46 ಇದ್ದು ಸೇರಿಕೊಂಡು ಲಾಸ್ಟಿಗೆ 23 ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗ್ತದೆ. ರೈಟ್. ಸೊ ಹಿಯರ್ ವಿ ಹ್ಯಾವ್ 46, ಇಲ್ಲಿ 46 ಅದಾವೆ, ಇಲ್ಲಿ 23 ವಿಲ್ ಮೂವ್. ಐ ಹ್ಯಾವ್ ಶೋನ್ ಯು ಓನ್ಲಿ 2, ಬಟ್ ಇವು ಬಾಕಿ ಎಲ್ಲ ಇಲ್ಲೇ ಇರ್ತಾವೆ. ರೈಟ್. Then what happens in the next phase? Fourth one. How can you explain anaphase? In anaphase 1, chromosomes start moving towards the opposite poles. The centromere does not split as a result after the contraction of the spindle fibers. Contraction antadra by the pulling action. Contraction of the spindle fibers, the chromosomes will move towards the opposite poles, and because there is no what? Cutting up of the centromere, or there is no splitting up of the centromere, the chromosomes are in dyad position. Dyad andre 2 chromatid arm iruvanthadu dyad. 4 chromatid homologous chromosomes ಆಗಿರುವಂಥದ್ದು ಟೆಟ್ರಾಡ್. 4 ಆರ್ಮ್ ಇರುವಂಥದ್ದು ಟೆಟ್ರಾಡ್ ಅಥವಾ ಬೈವ್ಯಾಲೆಂಟ್ ಅಂತ ಕರಿತೀವಿ. ದಿಸ್ ಇಸ್ ಎ ಮೋನೋವ್ಯಾಲೆಂಟ್ and dyad, 2 chromatid arms iruvantavu. They start moving towards opposite poles. That is the characteristic feature of the anaphase. What happens in the telophase? In the telophase, the chromosomes will move towards the opposite poles. Then after that a separate nuclear membrane is reformed. Then after movement there is a cytokinesis. That means the cell starts to divide with the help of a cleavage furrow. Then finally this cell, the furrow gets totally deepened to form into what? 2 cells. Finally it forms into 2 cells. 2 cells ಆಗ್ತಾವೆ. But the number of chromosomes here is n. N means half. It is a reductional division. The chromosomal number gets reduced to half here. Right? This is regarding the part of the telophase. Telophase is a condition where the chromosomes totally move towards the opposite poles and there is a karyokinesis. Nuclear division is there. So there is a formation of a nuclear membrane and a nuclear envelope. Nuclear envelope is formed, nucleolus is formed. So all the cell organelles are formed. Then after that the cell will cut into 2. That means karyokinesis is followed by cytokinesis. Cytokinesis is division of cytoplasm. It occurs. Right. By this the chromosome number remains half. That means from one cell, idr hinde iruvanthadu one cell irtada. Idu prophase. This is prophase 1. This is metaphase 1. This is anaphase 1. This is telophase 1. After these phases, you 4 phase ಆದ ನಂತರ 4ನೇ phase ಒಳಗಡೆ ಇಲ್ಲಿ ಕ್ರೋಮೋಸೋಮ್ ಇಲ್ಲಿ 46, here also 46, here also 46. ಯಾಕಂದರೆ ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನ್ ಆಗಿಲ್ಲ. ಇಲ್ಲಿ ಬಂದು 23 ಆಗ್ತದೆ, ಟೆಲೋಫೇಸ್ನಾಗ ಬಂದು 23 ಆಗ್ತದೆ. 23 ಆಗೋದು ಡಿಸೈಡ್ ಆಗೋದು ಯಾವ ಫೇಸ್ ಒಳಗಡೆ, ಅನಾಫೇಸ್ ಒಳಗಡೆ. ಯಾಕಂದರೆ ಎಷ್ಟು ಕ್ರೋಮೋಸೋಮ್ ಮೂಮೆಂಟ್ ಆಗ್ತಾವೆ ಅನ್ನೋದ್ರ ಮೇಲೆ ಡಿಪೆಂಡ್ ಆಗ್ತದೆ. ದಿಸ್ ಈಸ್ ವಾಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಎಸ್ ಡಿಸ್ಜಂಕ್ಷನಿಂಗ್. ಡಿಸ್ಜಂಕ್ಷನಿಂಗ್ ಅಂದ್ರೆ ಅಪೋಸಿಟ್ ಕಡೆ ಮೂಮೆಂಟ್ ಆಗ್ತಾವೆ. ನಾನ್ ಡಿಸ್ಜಂಕ್ಷನಿಂಗ್ ಅಂದ್ರೆ ಹಂಗೆ ಮೂಮೆಂಟ್ ಆಗಂಗಿಲ್ಲ ಅಥವಾ ಒಂದೇ ಕಡೆ ಮೂಮೆಂಟ್ ಆಗ್ತಾವೆ. ದಿಸ್ ಈಸ್ ಡಿಸ್ಜಂಕ್ಷನಿಂಗ್ ಆಫ್ ದ ಕ್ರೋಮೋಸೋಮ್ಸ್. ಈಕ್ವಲ್ ನಂಬರ್ ಆಫ್ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಮೂವ್ ಟುವರ್ಡ್ಸ್ ದ ಅಪೋಸಿಟ್ ಪೋಲ್ಸ್. ದೆನ್ ಫೈನಲಿ ಇಲ್ಲಿ 23 ಇಲ್ಲಿ 23 ಆಗ್ತಾವೆ. ದ್ಯಾಟ್ಸ್ ವೈ ಮಿಯೋಸಿಸ್ ಈಸ್ ಕಾಲ್ಡ್ ಎಸ್ ಅ ರಿಡಕ್ಷನಲ್ ಡಿವಿಷನ್. ಇನ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಇಲ್ಲಿ ಜನರಲ್ ವ್ಯೂ ಬೇರೆ, ಪರ್ಟಿಕ್ಯುಲರ್ ವ್ಯೂ ಬೇರೆ ಆಗ್ತದೆ. ಇನ್ ಜನರಲ್ ಮಿಯೋಸಿಸ್ ಇಸ್ ರಿಡಕ್ಷನಲ್ ಡಿವಿಷನ್ ವೇರ್ ದ ಕ್ರೋಮೋಸೋಮಲ್ ನಂಬರ್ ಆರ್ ರಿಡ್ಯೂಸ್ ಟು ಹಾಫ್ ಇನ್ ಈಚ್ ಆಫ್ ದ ಡಾಟರ್ ಸೆಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಬಟ್ ಇನ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಮಿಯೋಸಿಸ್ 1 ಈಸ್ ರಿಡಕ್ಷನಲ್ ಡಿವಿಷನ್, ಮಿಯೋಸಿಸ್ 2 ಈಸ್ ಇಕ್ವೇಷನಲ್ ಡಿವಿಷನ್, which is exactly similar to that of the mitosis, right? So here it completes the part of the meiosis 1. Illige meiosis 1 mugitu. Then from the same cell the meiosis 2 will start. But meiosis 2 start agಬೇಕಂದ್ರೆ ಅಲ್ಲೊಂದು short gap ಅದ, that short interval is called as intrakinesis. So the gap is called as intrakinesis. What is intrakinesis? It is an objective question. Intrakinesis is a phase where there is a gap between two meiosis. The gap between two meiosis, meiosis 1 and meiosis 2 is called as intrakinesis. Write the definition. The gap between two meiosis is called as intrakinesis. This intrakinesis is very short-lived. It is a very short-lived. That's why this intrakinesis is said to be a shorter stage, a smaller phase of meiosis 1 and meiosis 2. Then what happens? Then continues the meiosis. Meiosis 2 continue ಆಗ್ತದೆ. Now comes the part of the meiosis 2. Then meiosis 2 has how many phases? Again same 4 phases. Characters are exactly same which you have written in the mitosis. Mitosis ಏನು 4 ಫೇಸ್ ಬರ್ದಿರಿ ಅವ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಇಲ್ಲಿ ರಿಪೀಟ್ ಆಗ್ತಾವೆ. ಸೊ ಹಿಯರ್ ಮಿಯೋಸಿಸ್ 2 ಇಟ್ ಹ್ಯಾಸ್ ಹೌ ಮೆನಿ ಸ್ಟೇಜಸ್? 4 ಸ್ಟೇಜಸ್. ಫಸ್ಟ್ ಒನ್ ಇಸ್ ಪ್ರೊಫೇಸ್. ವಿ ರೈಟ್ ಹಿಯರ್ ಪ್ರೊಫೇಸ್ 2, ಇಲ್ಲಿ ಪ್ರೊಫೇಸ್ 2 ಅಂತ ಬರೀತೀವಿ. ಸೆಕೆಂಡ್ ಫೇಸ್ ಇಸ್ ಮೆಟಾಫೇಸ್ 2. ಥರ್ಡ್ ಸ್ಟೇಜ್ ಅನಾಫೇಸ್ 2. And last one telophase 2. Same phase, same characters, nothing change. ಇಲ್ಲೇನು ಚೇಂಜ್ ಇಲ್ಲ. ದ್ಯಾಟ್ಸ್ ವೈ ದಿಸ್ ಈಸ್ ವೆರಿ ಸಿಂಪಲ್. ಅದಕ್ಕಾಗಿ ಇದು ಸೆಕೆಂಡ್ ಪಾರ್ಟ್ ಭಾಳ ಸಿಂಪಲ್ ಅಂತ ಹೇಳಿದೆ. ಪ್ರೊಫೇಸ್ 1 ಮಾತ್ರ ಎಲಾಬ್ರೇಟ್ ಸ್ಟೇಜ್ ಇರುವಂಥದ್ದು, ಬಾಕಿ ಎಲ್ಲ ದೇ ಆರ್ ವೆರಿ ಸಿಂಪಲ್. ದೆನ್ ಲೆಟ್ ಸಿ, ಇಲ್ಲಿಂದ ಕಂಟಿನ್ಯೂ ಆಗ್ತದೆ. ವಾಟ್ ಹ್ಯಾಪನ್ಸ್ ಇನ್ ದ ಪ್ರೊಫೇಸ್ 2? During meiosis 1, when it reaches to the telophase 1 stage, during this telophase, then the chromosomes which are condensed will become decondensed. Decondensing ಆಗ್ತದೆ. ಒಳಗೆ ವಾಪಸ್ ತಿರ್ಗ ಈ ದಾರದ ಉಂಡಿ ಒಳಗೆ ಇರುವಂಥ ರೀಲ್ ಉಚ್ಚಿದಂಗೆ ಆ ರೀತಿ ಆಗ್ತದೆ. ಡಿಕಂಡೆನ್ಸೇಷನ್ ಅಂತ ಕರಿತೀವಿ. ವಾಪಸ್ ತಿರ್ಗ ಪ್ರೊಫೇಸ್ 2 ಅಂತ ಬಂದಾಗ ಅಗೇನ್ ಇಟ್ ಗೆಟ್ಸ್ ಕಂಡೆನ್ಸ್ಡ್. ಡಿಕಂಡೆನ್ಸ್, ರಿಲೀಸ್, ಕಂಡೆನ್ಸ್ ಟು ಬಿಕಮ್ ಶಾರ್ಟನ್. ಇಲ್ಲಿಂದ ಕಂಟಿನ್ಯೂ ಆಗ್ತದೆ. ಈ ಸೆಲ್ ಇದು ಪ್ರೊಫೇಸ್ 2 ಒಳಗಡೆ ಹೋಗ್ತದೆ, ಇದು ಸೆಲ್ ಪ್ರೊಫೇಸ್ 2 ಒಳಗಡೆ ಹೋಗ್ತಾವೆ. ಲುಕ್ ಎಟ್ ದಿಸ್. ಓಕೆ, ನಾವು ಹಿಯರ್ ಇಟ್ ಗೆಟ್ಸ್ ಕಂಡೆನ್ಸ್ಡ್ ಟು ಫಾರ್ಮ್ ಇನ್ ಟು ವಾಟ್? ಕ್ರೋಮೋಸೋಮ್. ದಿಸ್ ಈಸ್ ಪ್ರೊಫೇಸ್ 2. ಇಟ್ ಈಸ್ ಎ ಕಂಟಿನ್ಯೂಡ್ ಸೆಷನ್. ಈ ಸೆಲ್ಲು ಪ್ರೊಫೇಸ್ 2ದಾಗ ಹೋಗ್ತದೆ, ಈ ಸೆಲ್ಲು ಪ್ರೊಫೇಸ್ 2ನಾಗ ಹೋಗ್ತದೆ. ಹಾಗಿದ್ರೆ ಲಾಸ್ಟಿಗೆ ಎಷ್ಟು ಎಂಡಿಗೆ ಎಷ್ಟು ಸೆಲ್ಸ್ ಫಾರ್ಮ್ ಆಗ್ತಾವೆ ಇದ್ರ ಮೇಲೆ ನೀವು ಗೆಸ್ ಮಾಡ್ಬೋದು. ಓಕೆ, ಇಟ್ ಎಂಟರ್ಸ್ ಇನ್ ಟು ಪ್ರೊಫೇಸ್ 2. ಪ್ರೊಫೇಸ್ 2 ಈಸ್ ಎ ಕಂಡೀಷನ್ ವಿಚ್ ಆಕರ್ಸ್ ಆಫ್ಟರ್ ದ ಟೀಲೋಫೇಸ್ 1 with a gap called as interkinesis. After this interkinesis, in the prophase 2, the decondensed form of the chromosome gets condensed to form into chromosome. So chromatin thread gets condensed to form into chromosome. Then what happens? Again the process continues. ಇಲ್ಲಿ ಮತ್ತು ವಾಪಸ್ ತಿರುಗ ಕಂಟಿನ್ಯೂ ಆಗ್ತದೆ. They will enter into the next stage. Next stage is called as a metaphase. Now during prophase, what you have written in the mitosis prophase, same character. The nucleolus will disappear, nuclear membrane will disappear, chromosomes are present entirely in the cytoplasm of the cell. Then other cell organelles also disappear. ಇಲ್ಲಿ ಏನೇನು ಡಿಸಪಿಯರ್ ಆಗ್ತದ ಲಾಸ್ಟ್ ಸ್ಟೇಜ್ನಾಗ ವಾಪಸ್ ರಿಅಪಿಯರ್ ಆಗ್ತದ. ಪ್ರೊಫೇಸ್ನಾಗ ಡಿಸಪಿಯರೆನ್ಸ್, ಟೀಲೋಫೇಸ್ನಾಗ ರಿಅಪಿಯರೆನ್ಸ್, ಈ ರೀತಿ ಆಗ್ತಾ ಇರ್ತದೆ. ಓಕೆ, ನಾವು ಹಿಯರ್ when this prophase 2 will continue to the next phase is called as a metaphase. Now again in the metaphase the chromosomes are arranged at the equatorial plate. Look at this, the chromosomes are arranged at the equatorial plate. Then the centrioles will move towards the opposite poles. They produce the spindle fibers, and these spindle fibers will attach to the kinetochores of the chromosomes. Now tell me, how many chromosomes are there here? ಇಲ್ಲಿ chromosomes ಎಷ್ಟು ಅದಾವು? 23 ಮಾತ್ರ, 23 ಅದಾವು ಇಲ್ಲಿ. Then this side again same, ಇಲ್ಲೂ ಅದೇ ರೀತಿನೇ ಅದ. Here the chromosomes are arranged at the equatorial plane. The centrioles will move towards opposite poles. They produce the spindle fibers, thus completing metaphase 2. Then ಮುಂದಿನ process ಬರೀತದ. ನೆಕ್ಸ್ಟ್ ಏನ? Anaphase. ಓಕೆ, ಸೊ ನೆಕ್ಸ್ಟ್ ಒನ್ ಈಸ್ Anaphase. ಎರಡು ಸೆಲ್ Anaphase ಒಳಗಡೆ ಹೋಗ್ತಾವೆ. ದೆನ್ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್ ಇನ್ ದ Anaphase? ಹಾಗಿದ್ರೆ ಇಲ್ಲಿ Anaphase ಒಳಗಡೆ ಏನಾಗ್ತದ? ಲೆಟ್ಸ್ ಸಿ ಇಟ್. ಇನ್ ದ Anaphase the chromosomes start moving towards opposite pole. But how? In this phase, look at the phase here. So what are the names that we are going to give? This is prophase 2, this is metaphase 2. What happens in the Anaphase 2? Anaphase 2 ಒಳಗಡೆ ಏನಾಗ್ತದ ಅನ್ನುವಂಥದ್ದು. Now here the centromere splits. ಇಲ್ಲಿ ಸೆಂಟ್ರೋಮಿಯರ್ ಹೊಡಿದದ, ಯಾಕಂದರೆ ಸೆಂಟ್ರೋಮಿಯರ್ಗೆ ನಾಲ್ಕು ಕ್ರೊಮ್ಯಾಟಿಡ್ಸ್ ಅದಾವೆ, 1 2 ಕೆಳಗಡೆ 2 ಇರ್ತಾವೆ. ಸೆಂಟ್ರದಾಗ ಇರುವಂಥ ಸೆಂಟ್ರೋಮಿಯರ್ ಸ್ಪ್ಲಿಟ್. ಯಾವಾಗ ಇವು ಸ್ಪ್ಲಿಟ್ ಆಗ್ತಾವಲ್ಲ, ಅಟ್ಯಾಚ್ಮೆಂಟ್ ಆದಾಗ ಎಷ್ಟು ಕ್ರೋಮೋಸೋಮ್? 23 ಅದಾವೆ. ಯಾವಾಗ ಸ್ಪ್ಲಿಟ್ ಆಗ್ತಾವಲ್ಲ? 46 ಆಗ್ತಾವೆ. ದ ಸೆಂಟ್ರೋಮಿಯರ್ ವಿಲ್ ಸ್ಪ್ಲಿಟ್ because the spindle fibers will pull towards the opposite poles. That's why the chromosomes start moving towards opposite poles. Now look at this point. Now look at this structure. ಇಲ್ಲಿ ನೋಡ್ರಿ, ಇಲ್ಲಿ 4 ಆರ್ಮ್ ಇದ್ದಿದ್ದು ಇಲ್ಲಿ 2 ಆರ್ಮ್ ಆಗಿ ಬಿಡ್ತಾವೆ. ಯಾಕಂದ್ರೆ ಸೆಂಟರ್ದಾಗ ಇರುವಂಥದ ಸೆಂಟ್ರೋಮಿಯರ್ ಸ್ಪ್ಲಿಟ್ ಆಗ್ತದೆ. ಇದು ಸ್ಪ್ಲಿಟ್ ಆದಾಗ 46 ಆಗ್ತಾವೆ. 46 ಆದಾಗ ಅವಾಗ ಅರ್ಧ ಈ ಕಡೆ 23. ಅದಕ್ಕಾಗಿ ಲಾಸ್ಟಿಗೆ ಎನ್ ಆಗ್ತದೆ. ಅದಕ್ಕಾಗಿ ಪ್ರೀವಿಯಸ್ ಸೆಷನ್ ಒಳಗಡೆ ಒಮ್ಮೆ ಡಿಸ್ಕಷನ್ ಮಾಡಿದ್ವಿ. ಮಿಯೋಸಿಸ್ 1 ರಿಡಕ್ಷನಲ್ ಡಿವಿಷನ್ ಎನ್ ನಂಬರ್ 23 ಆಗ್ತಾವ. ಎನ್ ಆದದ್ದು ಮುಂದೆ ಇವ ಡಿವೈಡ್ ಆದಾಗ ಹಾಫ್ ಎನ್ ಆಗಂಗಿಲ್ಲ. ಯಾಕೆ ಹಾಫ್ ಎನ್ ಆಗಂಗಿಲ್ಲ ಅಂತಂದ್ರೆ ರೀಸನ್ ಈಸ್ ಸೆಂಟ್ರೋಮಿಯರ್ ಇಲ್ಲಿ ಸ್ಪ್ಲಿಟ್ ಆಗ್ತದೆ, ಸೆಂಟ್ರೋಮಿಯರ್ ಇಲ್ಲಿ ಸ್ಪ್ಲಿಟ್ ಆಗಂಗಿಲ್ಲ. ಅನಾಫೇಸ್ 1 ಒಳಗಡೆ ಸ್ಪ್ಲಿಟ್ ಆಗಂಗಿಲ್ಲ, ಇಲ್ಲಿ ಸ್ಪ್ಲಿಟ್ ಆಗ್ತದ ಸೆಂಟ್ರೋಮಿಯರ್. ಓಕೆ, ಹಿಯರ್ ಆಲ್ಸೋ ದ ಸೆಂಟ್ರೋಮಿಯರ್ ವಿಲ್ ಸ್ಪ್ಲಿಟ್. ದ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಸ್ಟಾರ್ಟ್ ಮೂವಿಂಗ್ ಟುವರ್ಡ್ಸ್ opposite poles. So this is the most important feature of the Anaphase. Finally, half of the chromosomes will move towards one pole, half of the chromosomes will move towards one pole. ಇದರ ಮುಂದಿನ ಸ್ಟೇಜ್ ಏನಾಗ್ತದೆ? ಓಕೆ, ಅದರ ಮುಂದಿನ ಸ್ಟೇಜ್. ಸೊ ಲೆಟ್ ಮಿ ಟೇಕ್ ಇಟ್ ಡೌನ್. ವಾಟ್ ವಿಲ್ ಹ್ಯಾಪನ್? ಫ್ರಾಮ್ ದಿಸ್ ಒನ್ ಸೆಲ್ Anaphase ಇಂದ ಏನಾಗ್ತದೆ? 2 cells are formed. From one cell ಇದರಿಂದ ಇಲ್ಲಿ ಆಗ್ತದೆ. It is N, this is N. From the next one is cell ದಿಂದ again we get 2 cells. This is N, this is N. Look at this. This is what? This is a telophase 2. Telophase 2, the karyokinesis is followed by cytokinesis. Like one cell is cytoplasm divide ಆಗ್ತದೆ. The karyokinesis is followed by the cytokinesis, where 4 haploid cells are formed. Remember, at the end of meiosis 2, 4 cells are formed. At the end of meiosis 1, 2 cells are formed. At the end of meiosis 1, 2 haploid cells are formed. At the end of meiosis 2, 4 haploid cells are formed. At last, the concluding statement is, during the process of meiosis, how many cells are produced? During the process of meiosis, 4 haploid cells are produced, and mainly they are gametic cells. You gametes ಆಗ್ತವೆ, either it is a egg or a sperm, right? So this is regarding the part of the meiosis. So with this we have the last part is regarding the significance of the meiosis. So we have the significance of meiosis. Meiosis ಇಷ್ಟೆಲ್ಲ ನಾವು ಕಲಿತೀವಿ, ಏನು ಸಿಗ್ನಿಫಿಕೆನ್ಸ್ ಇದು ಅಂತ ಅನ್ನುವಂಥದ್ದು ಲಾಸ್ಟ್ ಪಾರ್ಟ್, right? As we have already studied regarding the significance of the mitosis, now here we have the significance of meiosis. Meiosis ಎಲ್ಲಿ ಆಗ್ತದ? Meiosis occurs in sexually reproducing organisms. Then what is the end result, end product of meiosis? The end product of meiosis is formation of gametes. Gamete form ಆಗ್ತಾವೆ ಇಲ್ಲಿ. Okay, that is the end product, right? So let's see the significance of meiosis. Okay, in the last part, in the significance of the meiosis. So meiosis, it occurs in sexually reproducing organisms. So it occurs in sexually reproducing organisms. Okay, so this is the character. Then it leads to formation of gametes. It leads to formation of gametes. So finally what are formed? Gametes are formed. Then by this it brings about a genetic recombination. It brings genetic recombination. How? I'll tell you. Then apart from this, finally these variations helps in evolution. These variations helps for evolution. So this is regarding the significance of the meiosis. Idu meiosis significance ಅಂದು ಕರಿತೀವಿ. It occurs in sexually reproducing organisms, where finally it leads to the formation of the gametes. So gametes has to be formed, egg and sperm has to be formed, or ovum and sperm has to be formed. That's why it occurs in sexually reproducing organisms. And remember, meiosis occurs in which cells? Germ cells and karitivi. Germ antadre sex cells and karitivi. Mitosis occurs in the body cells, where it is called as a somatic cells. You can add that character also. Meiosis occurs in germ cells, right? Mainly our germ cells and bandaga, in the males it is called as spermatogonia, in the females it is called as oogonia, which you are going to study it in second PUC. Okay. Then it brings about genetic recombination. Genetic recombination ಆಗ್ತದೆ by overlapping of the non-sister chromatids, which already you have studied in the third stage, pattern stage. This is called as recombination or crossing over. Recombination is nothing but a genetic exchange of the material, genetic exchange of the DNA. Okay, so gene recombination ಅಂತ ಕರಿತೀವಿ ನಾವು. As there is genetic exchange in between two homologous chromosomes, it brings about what? Variations. ಇದರಿಂದ ಬದಲಾವಣೆ ಹಾಕೊಂತ ಬರ್ತದೆ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಒಳಗಡೆ. ಕ್ಯಾರೆಕ್ಟರ್ ಒಳಗಡೆ ಚೇಂಜ್ ಹಾಕೊಂತ ಹೋದಂಗೆ ಹಂಗೆ ಮುಂದಿನ ಜನ್ರೇಷನ್, ನೆಕ್ಸ್ಟ್ ಜನ್ರೇಷನ್ ಅಂದರೆ 5 ಜನ್ರೇಷನು, 10 ಜನ್ರೇಷನು, 20 ತಲೆಮಾರು ಇರ್ಬೋದು, 30 ತಲೆಮಾರು ಇರ್ಬೋದು, ಹಿಂಗೆ ವೇರಿಯೇಷನ್ಸ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಹಾಕೊಂತ ಹೋಗಿ finally there is evolution. Evolution is nothing but formation of a new species. That means we are human beings. By us, in few generations, superhuman beings are going to evolve. That is called as evolved animal. That is what is termed as evolution. ಅದಕ್ಕೆ ನಾವು evolution ಅಂತ ಕರಿತೀವಿ. Regarding the process of evolution, you are going to study it in the second year. Evolution ಅನ್ನುವಂಥ there is a big chapter where you are going to study it. But this is the significance. That means the law of nature is change. Change is the rule of the nature. As a result, as there is a change in the genes, finally these change in the genes leads to variations. Variations ದಿಂದನ evolution ಆಗ್ತಾ ಇರ್ತದ. So let's continue the last session of the question part, neat questions, and with that we'll end up the chapter. So till then, watch our videos and thank you.